I never forget when in the early 90s, when I was taught immunotherapy in Pittsburgh. My teacher, Mike Lowe, said, well, cancer is nothing than a bag of mutations. It's awful. It goes haywire, and it's different, and sometimes there appears to be randomness, and sometimes there are commonalities. And I like to get some feedback from you. What is the difference if we look at cancer cells in ourselves and our normal cells? How are they very much different, or are they pretty similar? Who is for pretty similar? Wow, good, great, I love it. Yes, because this is, they are pretty much, even if they do look growth. If you look at the chromosome paintings, the little sausages that were shown, there are all sorts and forms, and there are deletions and things are inserted. It's like you would read a text and you don't understand it. However, if you go back and look at the entire picture, it's pretty much similar. They're still our own cells. They're still part of our immune cells. So an anti-immune attack is, in nature, a very focused autoimmune response. It's part of ourself. It's not an alien living in us. Second question. If I compare, it's about individuality. If I compare my DNA, and I don't say that I'm my DNA. So we are made up by many things, by life exposures, and so it's not our mind and our soul, but our DNA blueprint. What makes us different? What tissue? If I say, well, I remove that tissue, and then this gives us the most significant difference between you and me, what would you take out? Or are we pretty similar? The easy answer is actually the immune system, right? Because we are 99.9%, we're all, it doesn't matter if we go to Australia or Japan or back in time. The only thing that makes us different, you and me, is the diversity in our immune system. And this is shaped by a lot of things, by infections, by exposures, by randomness, by awful things and good things that happen to us, the bacteria that live within us, and so on. So the bag of mutations, the strange stuff that happens in a tumor, is supervised and hopefully recognized by the immune system at the other side, which makes us unique. My immune system makes myself unique. If I compare the genes that are non-immune related, we are pretty much identical in this room, except those who have or had cancer, and most of us actually had cancer, but it was eradicated by the immune system. Now, this is the Champalimaud Foundation. That's the Center for the Unknown, and this is where Vasco da Gama put the ship, the boats in the ocean to sail to the India. That's the reason why we go to the unknown and try to find out actually new horizons. So that's the interplay, and this is the laser pointer. Let's see if this works.
The laser pointer should work, yeah.
No?
Something switch. Might need.
Oh.
Oh.
Okay, doesn't matter. So essentially what you have here is that there's a tumor and there's an entire interplay of immune systems. And there are cells that, they're chauvinists in life. There are two kind of immunologists. Some are more for the more educated, the more stylish immune cells. We call them adaptive. The adaptive immune cells, the CD4 cells, gamma delta cells, killer cells, helper cells. They have receptors. Thank you very much. They have receptors here. And so they recognize the random mutations. They apparently recognize all the things that went wrong. And there are these cells that are it's a certain breed of doctors and researchers. They're more attracted to the neutrophils and macrophages. There's essentially the bullies, the attackers. Well, you bad boy, go away. You bad. You don't belong here. The flavor of you is not. You don't belong to the normal cells. I can't tell exactly why, but you're a cancer cell. Here it's much more refined. Well, you have 1 point mutation, and you don't fit here. Here it's much more, we call it innate. It's much more raw. It's a recognition that you don't belong. And then, so this is believed to be a first wave. That's an interplay. One does not exclude the other. So there's this innate response, the natural killer cells, the new cells that have been identified, so the textbooks are constantly rewritten, and are cells that are adaptive. They recognize very fine mutations. And have you heard that we got one chromosome from the father, the other one from the mother, and we have a certain genetic background. This helps us. This puts us at risk, maybe to develop cancer, maybe to have some sort of an autoimmune disease. For instance, if you have narcolepsy, this happened in Scandinavia, for instance. If you have a certain genetic background, HLA-DQ0602, that's a sort of a structure on the cell. Then if you received the influenza vaccination 2009, if you're very excited to fall asleep. Not good. So you actually, there were some bankers in London and in Frankfurt, whenever they were very excited, they really had to sort of think about some exit ways actually to go in the room before they get excited, because it didn't do great actually when they're falling asleep and great things happened. So the point is that there is a genetic background that helps the immune system to recognize. So, which means even if you have mutations, they may not be seen by the immune system. And we know this from a number of diseases. For instance, HIV, there's so-called the long-term non-progressors. And we know if you have a certain genetic background, then the immune system can see beautifully certain mutations of the virus, and they keep the virus at bay. And we have a similar situation in cancer. Now, there are different ways to fight cancer, and we discuss some of them. Some of them are here. TIL, tumor-infiltrating lymphocytes. This is a treatment where the tumor is taken out, and there are immune cells, the innate ones, which we tend to ignore, but they are as well important, and the adaptive immune cells, the CD4 cells, the killer cells, and the helper cells, CD4 and CD8 cells. And they're not there for just by chance. We say, well, they may not be very helpful because they lost a battle, but we can see it from a different point. We say, well, there are the soldiers in the trenches, and they are as close as it gets to the enemy, but they lost it. But if we have just 100 soldiers, only if they're few, if you make an army of them outside the body, they know the enemy best, and they're able to recognize all the mutations and the craziness that is going on. And then we can use them. This is exactly done in these TIL. So the tumor is being excised, is being removed here, and there are growth factors, and the TIL, it's called enrichment, they're grown to 10 to the 10th cells, a bag of cells that is being reinfused, or essentially then a different possibility, go down here, and say, well, the T cells in the tumor didn't do a great job. Maybe they don't recognize if we have the ocular melanoma, if we have a liver metastasis, if we have the metastasis somewhere else. So why do I know that T cells removed from the liver recognize metastasis in the lung, for instance? So let's go to the blood. The blood is essentially the highway system of the immune system. So the T cells travel in the blood, the immune cells. There are only 2% of our immune cells in the blood at a given time. 98% do job somewhere. It's a traveling. It's just like if you say, well, how many Mercedes or Renault or Audi are in London, and you would just look at the street or go, and essentially it doesn't say how many Mercedes really are present. So, but if you just observe long enough, you may have a representative idea. And this is essentially what's going on here. So immune cells are taken from the blood and they are tested whether they recognize the mutations, the craziness, the randomness that is going on in the tumor. They are educated to fight better, and those that are against the tumor are enriched and giving back. And there's a third possibility. I said, well, it did not work. Why should we why should we waste time to take T cells from the tumor or from the peripheral blood? We don't take a risk. We use a T cell receptor, that's the recognition molecule, from one patient who was able to fight cancer and has a full response. We don't know if it's cured, but essentially these cells killed the cancer. And if we use the same receptor for a patient with the same genetic background, we can genetically engineer it. So why care? So we just transfer this receptor into the patient's own cell and endow the patient with a specificity that may not have been present before. And this is a T cell receptor that recognizes, for instance, mutation, or a CAR. It's a chimeric antigen receptor. A chimera in the Greek history is essentially two creatures that don't exist, but essentially they're fused to each other. And that's a B cell. B cells are the immune cells that make antibodies. You have an antibody attached to a T cell. So an immune cell would see the cancer cell and then give a signal, kill. So these are the different forms of cellular immunotherapy to get the T cells out from the tumor because they know the enemy, make it fightful, make it T cells on speed, enrich them, or you take the few fighters that are traveling in the blood, or you say, well, this may not work, and you transfer genetic information, a blueprint from patients with cancer who are able to fight the disease with a genetic background, and these cells are giving back. And this is what we call cellular therapy. There's a number, and I can skip that, of a lot of immunotherapies going on, particularly in melanoma. And this is essentially a different way of immunotherapy. You take out the brakes of the immune cells, because otherwise we almost would end all up with autoimmune responses. Everything which is good has to come to an end, right? It would be awful if you have a skin infection. It would just go on and go on and go on. And this is sometimes what happens in tumors. It's essentially a wound that never heals. So inflammation can drive immune responses. And inflammation can drive also the expansion of T cells that are not very helpful to fight cancer. What is checkpoint inhibitors do? They take away the brake from the helpful T cells that are able to recognize the chaos and the mutations. Now, let's just look what's going on in the tumor, and there are what we call, it sounds very fashionable, parallel profile of immune infiltrating. We already heard that cutaneous melanoma, skin melanoma, is different as compared to ocular melanoma. And if we really go into it, and this is not only a scientific exercise, this essentially has to do with really what we may be able to offer in immunotherapy. These are the killer cells. This is the kind of the brake system. Don't do anything. Cutaneous melanoma here, a lot of killer cells, but this brake system here. So, if we inhibit that, then these cells go crazy and kill. But this appears not to be present here in ocular melanoma, at least not in most lesions that have been taken out from patients. So this may also tell us that there's something unique immunologically. And this was a very practical study. It's like, can we grow these T cells out from a tumor and expand them to give them back to patient? Uveal melanoma, successful about 45%, 64%. And these are older numbers, but it appears to be, they can be grown. And I'm sure we can even crank this up to 90% now with the current technologies. So we can grow these T cells out. this was the pioneer of immune cells. Stephen Rose is a surgeon at the National Cancer Institute in Washington, the search for cure. This was in the mid-'80s when he infused for cutaneous melanoma T cells grown from melanoma lesions, expanded, grown in a hormone, a growth factor, IL-2, and then infused it back. So this is how it was. The tumor is being excised, and then there are different cells, and these different colors have a different meaning because, let's say, each color corresponds to a different genetic mutation. They are amplified, and then some of them may recognize the tumor. They are expanded and then giving back to the patient. That needs to be properly prepared. These are actually these are cells that we grew from a patient with uveal melanoma, and so there are these very, very active little guys here, and this is one of these T cells recognizing a tumor is attached to it, and they grow out in billions from the tumor lesions. There's the first patient who responded, still alive, treated in the 80s with cutaneous melanoma, and these T cells are grown in these big bags now and are being reinfused back in the patient. This was the 1988, the first report here. And this is what we see. These are lung metastases. So these are the spots that you here see 85 and 87. They are gone. The same is true with liver metastases here. And after infusion, they're gone. There was an important trial recently published from the Rosenberg group for patients with uveal melanoma. And essentially that's the same, with the same purpose. Get the metastasis out, grow the T cells, prepare the patients, and give it back. So we're very, very excited and looking into what was happening. No response, no response, no response, no response, cannot be evaluated. Partial response, complete response, no response, partial response, partial response. So it appears to be the response as compared to what we see in patients with cutaneous melanoma is pretty low, and some patients, and some individuals say, well, this is due because the mutational load is very low. This may only be partially true, but we see patients still have a complete response. Some have a partial response. So it is possible. Now, what do we see here? When Stephen Rosenberg expanded these T cells, this is what we see here, these bars is essentially how much tumor reactivity presents. It's as defined by production of gamma interferon. It's a hormone that kills tumor cells. And it appears that these T cells were able to recognize the patient's own tumor cells very well. This is how they're grown actually now in these big bags. We saw this. And I like to spend the last 3 to 5 minutes actually to see, what may have happened and what can we do different and what is the lesson learned. And this is essentially has to do with the genetic mutations. So usually what we do is, it doesn't matter if we have patients with glioma or pancreas cancer, patients with cutaneous or uveal melanoma, we say, well, we don't know. Really, I mean, there are these commercial tests. There are these mutations, what we call trackable targets. We say, well, it's a bag of mutations. We don't know what happens. Let's sequence everything. Everything. We call it tumor DNA exome sequencing. The exome is what translates the DNA in proteins. It's only 1% of our entire DNA information that we have, and then we know exactly the number of mutations. We can test them. So we know the enemy. We can test if these mutations are being recognized by the patient's own immune cells. Now, these are essentially, that's such a tumor clump. And what you see here is different mutations, genes, POTI, INSAM, whatever. I mean, so different genes here. And that's the wild type. This is why I say, well, it could be an anti-tumor response, could be an autoimmune response. And in different colors, you see the production of an interferon. This is something good, because this could kill tumor cells. this is a very nice tumor antigen because the T cells recognize only the mutation and not the wild type. But in red, there are only some tumor cells that express. So there is also a diversity in the tumor. Not every tumor cell has the same mutations. Now, where should we look? If we look in the T cells, the TIL that are in the tumor, there are very nice mutations that can be used. And if we look in the peripheral circulation in the blood, that appears to be very, very strong mutation because it's also vividly recognized in peripheral blood. If we look here in the T cells infiltrating into the tumor, it appears they can't do anything. They lost a battle. Each number is a mutation. But there is a vast knowledge how the tumor can be recognized in the peripheral blood. This is coming out now soon in the British Journal of Cancer, because we looked at where should we look? So we looked in the blood. There are some mutations that are only recognized in T cells of the blood, some only in the T cells from the tumor, and some in both. So there may be, let me see if this works, because that's what you forced me at 2 o'clock in the morning to look for this GIF, because eating the crumbs. Let's see that if this works here. Does not work. This is the Mac thing. Or is anybody able to help me to see if you have to push a button in a Mac that this kind of starts?
It's not meant to do it, is it?
Probably not. However, what you may have seen is these are tumor cells, and these little fellows here are T cells, and they are directed against mutations. And what you would see that these little fellows, they swim towards the tumor cell, and this is tumor cell. This is already kind of dying, and these 3 fellows attack the tumor cell. You see then the tumor cell shriveling up, and this is done in about 45 minutes. Then they're gone. And this is really eating then that's what we see here ex vivo. Why doesn't it happen in the patients? Why doesn't it happen in the people that we would like to treat? Location. This is a patient actually from Lisbon who was treated in the Frankfurt Ludwig Institute for Cancer Research, a patient with pancreatic cancer, very bad prognosis. And we had the T cells isolated from a metastasis from the primary tumor, and also from a patient got a colon biopsy because he had infection. What you see here is, this is not a boring slide actually, because essentially we looked at each of the patient's mutation. So before, it's not just a few scientific dots and everybody falls asleep. It's very exciting because what you see here is we have put mutation number 1, mutation number 2. This makes the cancer of this patient to her cancer. If she wouldn't have this 146 mutations, most likely, she would not have cancer. And we were able to test, what we can do for every patient, to see what kind of mutation is recognized by her T cells. If we get these T cells from the primary tumor, or from the metastasis, or from some different location. And essentially what we saw here, so the primary is green and the metastasis is in red. So there was nothing recognized, and suddenly, strongly recognized in the primary individual mutations. And look at this. A lot of mutations recognized in the primary tumor. What we infused into the patient was the T cells from the metastasis. And this is what everybody does. That's the standard, because what we think in medicine is like, okay, that's the metastasis. That's where the battle happens. That's her disease. That's the freshest thing that we can get. But it did not contain the T cells that were able to recognize her mutations. The primary tumors and are often seen as, that's the lost battle. And we see more and more that the T cells that we get from the primary tumors, so the location of these tumors are important. There's something very, very important in uveal melanoma. And I like to ask this. I did like to ask when I was more active in the field, to ask students actually that question: what is different from an electrical standpoint in uveal melanoma as compared to all other cancers? The loss of expression of a locus of one or more lens is a common phenomenon. Blah blah blah. And what does it mean? It means the following. Usually it's a disaster if the MHC molecules, that's the stuff that all these beautiful cells that I talked about, that we infuse, and the NCI trial was, is when these cells are fused, they can only see if the so-called transplantation engines, the MHC molecules, that tells the immune system, hitting the flag, there is a tumor, or there is a bacterial infection, or there is a virus there. If these molecules are lost, the mutations are still there, the virus is still in the cell, but the T cells don't see it. They are blinded. Usually it's a disaster, but not in uveal melanoma. When these molecules are left, patients appear to do better, have a better prognosis, and also as the disease advances. So we treat the patients as if they would have these molecules, but they lose them. So I think we have to look better at the landscape and really see what we do and what kind of... so the CD8 positive T cells and the CD4 positive T cells are all dependent on these MHC molecules. That's the lesson learned, and I'll never forget this. It was a patient I treated in Pittsburgh, 1987, melanoma, cutaneous melanoma. The patient had everything there. The tumor was controlled and recognized. And then what you see, this red color here, is lost. The tumor lost a molecule that makes it visible to the immune system. And the patient died within 4 weeks of an immense expansion of the tumor, and we put then this gene back with a retrovirus, and it was again recognized, of course in vitro. But then, you know, there's a song from Simon Garfunkel, 50 Ways to Leave Your Lover. And there are many ways of the tumor to make the immune system shut down. But there's also 50 ways of the immune system to recognize it. It's always smarter, and we have to help it to be smarter and facilitate it. That's a patient that we treated with pancreatic cancer, with metastasis to the lung. And we looked at lesions that were, I think it totally gave up the spirit here, there were different, that's a regressive, which means it was shrinking. Then there was a metastasis from the pancreas cancer lung was stable and peritoneal. And we look at different T cell subsets, the naughty T cell subset. But essentially, there was one was very interesting. This was shrinking. And it had so-called gamma delta T cells, very unusual cells. They're also even in fishes. They also have them. They're very old rudimentary cells. And apparently, this tumor lost MHC, they lost these antigens. And it was still recognized, and that's why the metastasis was melting away. And we looked at these cells, so we have each dot is a single cell, so there was a strong expression. This is also, you know, in colorectal cancer, so each cell here, and the brown staining is our MHC molecules. So everything is visible to the immune system. So it's like strong expression. And these are cancer cells. And they lose it. We can do whatever we want. We can vaccinate. We can give checkpoint inhibitors that will not be recognized by conventional CD4 and CD8 positive T cells. They will not. There are other molecules that are being recognized by immune cells, but in the course of the tumor, they will also disappear. So this CD57, this is MHC, class 1 molecule-like, is being also lost during tumor progression. But there is hope, because there was a recent report that the checkpoint inhibitors do not only excite cells that are defined cells and recognize each single mutation, they also activate the rogue cells. They say, well, you don't belong here, you don't sniff right. You know, you don't have the taste of being okay. And these are the natural killer cells. They go through the body and say, well, maybe virus infected you, maybe tumor. I don't know exactly what the mutation is, but you're wrong. And the checkpoint inhibitors also evoke. So into tumor escape goes into the tumor cells that are, even if they lose PD-L1 or PD-1, this breaks, the NK cells are also activated. So the last slide is the map targets and disease status. A cancer is not a cancer. The cancer at the time of diagnosis is a different cancer as compared to 3-4 months later, or a cancer after different courses of treatments, chemotherapy or immunotherapy. There is also a heterogeneity. A cancer in the lung, a metastasis, is different as compared to a metastasis in the liver. We have different instruments, like a surgeon. The CAR cells, with the T cell receptor, or we have with an antibody attached to it, the NK cells, B cells, vaccination, or CD8 and CD4 cells. And if the MHC molecule is lost, then the rogue cells may come in, the gamma delta T cells, or the NK cells that still recognize tumor cells, what they are. And this is where I like to end here. So we have our collaborators in Frankfurt and all the people here at the Champalimaud, and we think how, this is the reason why I called it immunosurgery, how the surgeons can take out certain cells or tumors, and this is also how our immune system works. And I think we can also instrumentalize the immune system, that we don't end up with autoimmune responses, but we can train it with similar instruments, like a surgeon, to take out the cells that are going haywire and doing the wrong things. Thank you very much.
Thank you. That was excellent. Any questions?
Thanks for that, Markus. I know we talked about, you touched on TILs and so on, and we know that with the trial in the States, the Ocular trial, 7 out of 20 people had a response, and I've come across a couple of them and they're just completely cured.
All right.
I've heard there's going to be an update to that, where he's going to take the T cells out,
Yeah.
and then do something really clever with them in an interim thing, and
Yes.
be selective with them, and
Yes.
then re-inject them. Do you know anything about that, or what that might entail?
Or very easy. Essentially, it's essentially looking at each tumor is being sequenced. So what I showed to you from the patient with pancreatic cancer, exactly what's happening at the NCI, right? So you have 150 mutations. And so what is grown from the patient's tumor is not only one TIL population, because it's different areas in the tumor. Let's say you have 20 areas. You have 20 different infusions that you could give. And you test each individual potential infusion against 150 mutations. And you're able to do two things. First, to say, well, I infuse only that population that shows the best reactivity to most of the broadest repertoire of mutations. Number one. Number two, I could even train them. I could use these tumor mutations to enrich and to make T cells even grow better and essentially expand, so we have them in higher numbers. So they are very educated and T cell and mutation enriched products that go back into a patient. Yeah.
Thanks for the wonderful presentation. So the lack of recognition by the immune system may be by deficient gene expression of the T cell receptor, right?
Usually, I mean, there are two different ways why it may not work. One is you have here the MHC molecule. So we have 6 from the father, 6 from the mother, and then we have the differently wired T cell receptor repertoire. So what usually happens that the MHC molecules that show and present mutations to the T cells that are either lost on the short arm of chromosome 6 or they're downregulated, that is the most common form to blind the immune repertoire. But you always need 2 to dance. Even if this is present, the MHC molecule, we need to have a proper T cell receptor recognizing the tumor mutation. And essentially there could be that a patient or any of us has not the T cell receptor repertoire because we may not have been able to sort out the right T cells. Maybe we had an infection in earlier life that just got rid of these T cells, or the tumor was boring out our T cell receptors. And there are 2 different ways. There's 3 different ways to overcome that. Number 1, that we, with vaccination against mutations, are able to expand and excite T cells that have not been expanded. Number 2, we say, well, in this T cell repertoire, in this patient's immune cell system, there are no proper T cells that are able to see these mutations. That's the reason when gene therapy comes in and will endow the T cells with a T cell receptor capable of seeing the mutations. And number 3, this is something very exciting. This is essentially a very pioneering work from Ton Schumacher in Amsterdam in the Cancer Center. He says, well, why not from other donors? Individuals that are genetically identical, but have a healthy immune system, that didn't go to chemotherapy, that didn't have a tumor yet, and we train the T cells from the donors that are 100% matched to see a mutation, and we give these T cells. A similar thing, actually, this is what happens a little bit in in stem cell transplantation. So we have thousands of people already mapped in our healthcare system, and we would be able to do these matches, and it takes advantage of these matches to supplement T cells that may not be present in a patient with cancer. So there are already, and this is not just nice theories, there may be a phase 1 clinical trial starting soon. You may look at the website from Ton Schumacher in Amsterdam.
So the cells are trained. That's how vaccination works. If the immune cells die over the time, how do they keep the information over the years? MicroRNA?
No, I mean, the question is who dies all the time? Because, I mean, we know from patients with cancer, and we know from patients with infectious disease that T cells can live on 10 years or even longer. It really depends where the hiding places are. Number 1, one of the most likely hiding place is the bone marrow. And since it's not very fun to have a needle in the bone marrow, we don't know very well actually the hiding place of tumor-reactive T cells, but it appears to be the case that long-term memory is not somewhere in the body, but in the bone marrow. This is, and we have very limited information about that. Another hiding place is what way, when immune cells are reacting, and one cell can kill 10 cancer cells in a short period of time, and then they get pretty exhausted. Usually they die. But then if you killed 10 tumor cells, let's say in 2 days, Some don't die, and these are the precious ones because they did a good job, and we would need them for long-term memory. Now, there's something particular happen. These are so-called immune cells with stem cell-like features, which means they have they're endowed with self-renewal. That's the fountain of youth. This is cells that can hang around for a long time. It appears that a minority of these cells who are so experienced are able to travel, so-called back in time, and pretend, or they look to us as if they would be innocent and fresh and youthful, but they're antigen experienced. And this is also a place where they reside. We can assume that a major percentage dies, but an important percentage may really either go in the bone marrow and or go back to these cells that we thought are only naive. This is where long-term memory cells reside now.